other terms, and we're going to look at those right now. So here we go. The vascular tunic is also composed of a ciliary body. Say it after me. Ciliary body. One more time. Ciliary body. Right? The ciliary body is the thickening ring of this choroid. Right? The choroid is transformed into a ciliary body as well as the iris. And we're going to talk about the iris in a second. But ultimately, the ciliary body is a muscle. Okay? We'll call it the ciliary muscle. Right? And the ciliary muscle ultimately changes the shape of the lens. It helps the lens. So it says it encircles the lens. That's where you can find it. But it helps the lens change shape. So let's just make sure that we are clear on that. So we're going to put that text down here. Changes the lenses, we'll add an apostrophe, shape to focus our vision. That's what the ciliary body does. Right? The ciliary processes in particular. Right? The ciliary zonule it just attaches the ciliary body to the lens. Right? A ligament, just like other ligaments attach. They're connective tissues. Okay, here another interesting diagram. Okay, we're looking at this particular part of the eye right now. All right, so again, let's orient ourselves. We see the cornea pointing over here. All right, we see the sclera connected to the cornea over here. Okay, so that makes up our fibrous tunic. Now moving inward, all right, we see the ciliary muscle, right, all down here, and the ciliary processes connecting the ciliary muscle to the lens, and the lens is this thing, right, and it can change shape. So again, the ciliary processes, the ciliary muscle, all of those make up the ciliary body, and the ciliary zonule connects it. Okay, and we don't really see the choroid over here, but if we were to imagine, the choroid would be somewhere in the middle here. Okay. So let's talk about the iris. If we are not already aware, the iris is the colored part of our eye, right, and it surrounds the pupil. The pupil itself is just an opening, right? It's a hole. It's the space where light can enter our eye. And the point of the iris, it's attached to the ciliary body as well, and it's composed of muscle. That's weird. If you hadn't known that, it might sound weird. But the iris, the colored part of your eye, is composed of muscle. And it's meant to constrict or dilate the pupil. So the point of the iris is to change the size of the pupil. Let's make sure that we have that down here as well. Right? The iris changes the shape or the size of the pupil. It's a muscle. Oops, if I can't spell. That'd be nice. That's not helping me out. I'll just erase. Of the pupil, period. Alright, there we are. The iris changes the shape or size of the pupil. And so the iris has really two smooth muscles. I'm not gonna ask you to differentiate them, but there's one of them called the sphincter pupillae muscle, which is the constrictor. It makes your pupil smaller, and the dilator pupillae muscle, which makes your pupil get bigger. Again, the iris acts to vary the size of the pupil, or changes the size or shape of the pupil. We've already tested this part of this anatomical structure of the eye already when we shined light into our eyes and we saw the pupil get bigger or get smaller. We saw it constrict and we saw it dilate. All right. And again, that's the job of the iris. Okay, and the last tunic, or the last layer that we'll talk about today in the eye, the deepest layer, is a layer we're going to call the sensory tunic. And as the name implies, the sensory tunic's job is to actually do the sensing, to actually receive information. And the part of the sensory layer that is really the most important, the most vital, is the layer that we're going to call the retina. And I think we've heard of this term before. The retina is the actual part of the eye that receives light and transmits it into a nervous signal, right? Into an electrical signal, essentially, that goes to our brain so that we can process it. So we're going to add a text box, and we're going to say the retina's job is to receive light signals and to convert them into, let's say, a nervous impulse that can be interpreted by the brain. Oops. I don't think I spelled that right. Interpreted by the brain. I know that's difficult to see. I'm going to change it in a second. Okay. Period. Get it off. All right. Oops. And make it bigger. There it is. All right. And let's change the font color just so that we can emphasize it here. Make it red. All right. The retina's job is to receive light signals and to convert them, and I know I spelled that wrong, but I'm not going to change it now, and to convert them into a nervous impulse that can be interpreted by the brain. Okay. And the retina itself is composed of two layers. Number one is going to be the pigmented layer. And again, like the choroid, the pigmented layer is so that light is not scattered. It does not escape the eye. We're really concerned, obviously, with the eye. We're concerned about keeping in that light so it doesn't escape, and so we can use as much light information as we can. The more important, I would say, layers we're going to call the neural layer. And the neural layer is a sheet of nervous tissue that contain or that are composed of more than one kind of neuron. All right, and we'll talk about one of these neurons in great depth. We're going to be talking about the photoreceptor cells. Okay? The photoreceptor cells are cells that receive light. Right? Photo is a prefix that means light. So we're just going to say these are cells that receive, oops, man, I really can't spell right now, receive light information. And the other cells are cells that convert this light in certain ways. One of them are called bipolar cells because they have two sides to them. All right, they're actually a relay station between the ganglion cells and the photoreceptor cells. We're going to see this in a second. All right. So again, bipolar cells have two ends. That's why we call them bipolar. Bi meaning two, poles meaning ends. Okay, let's move it. Oops, two ends. Man, I'm having a hard time typing, guys. And then the ganglion cells. We've talked about what ganglion means before. It's simply a bundle of cell bodies. So let's take a look at this. Right. We're looking at the microscopic anatomy of the retina, meaning we're looking at the cells. We're looking at the particular cells that we would see maybe under a high-powered microscope. And let's just take a look. Here are the ganglion cells. In fact, they are the anterior-most cells, okay? Because we're looking here from this perspective, and we're looking at it from this angle. So we're looking at it from the front. This would be the front over here. And the pathway of light, obviously light is going to be hitting the retina over this way. Okay, so the ganglion cells are the first cells that receive this information, and they'll transmit this information onto the bipolar cells in this layer over here, ultimately transmitting their information to the photoreceptors. Those are the three cells that I want you to pay attention to. I might ask you, in which order, from anterior to posterior, might you find these three kinds of cells? And it's your job to know that. All right. As you can see, and we'll talk about them in a sec, There are two kinds of photoreceptor cells. There are rod cells and cone cells. I think I know I had when I first learned about the eye, but I've heard, I think we've heard the rods and cones, or the term rods and cones with reference to our eyes. They refer, of course, to these photoreceptor cells, the cells that receive this light information. Okay, there are two, obviously, these rods and cone cells are the important cells. The rod cells are actually more sensitive to light than the cone cells. However, the interesting thing about the rod cells, even though they're more sensitive, they're only sensitive to black and white light, or rather to a grayscale. So we'll just call it black and white light, or black and white colors rather. So the rod cells, even though they are more sensitive, and I'm going to point an arrow to that. The rod cells are more sensitive to light, but they're only sensitive to certain colors, black and white, which obviously allows for vision in dim light, such as nighttime. Right, so your rod cells are very active when we're looking into the dark, or looking when it's dark. Cone cells are different. They operate best in bright light, and they are sensitive to all kinds of color vision. So when we have a very acute sense of color vision, and it's because of the concentration and sheer number of cone cells that we have in our retina. Right? And both of these cells, both of these photoreceptors are considered neurons. All right, let's take another look at them. All right, and they're called rod and cone cells simply because of their shape. Obviously, they're not perfect rods or not perfect cones, but the rod cells look a little bit more like straight rods, whereas the cone cells come to a pointed edge to them. And again, they receive light signals. We don't really need to know much more than that over here, and we're going to stop. We're not going to stop there. This is going to be our last slide, actually. On our retina, we're going to say that there are specialized regions. That's why we call this the regional specializations of the retina. And I'm going to start, actually, with the fovea centralis. Please say it after me. Fovea centralis. And one more time. Fovea centralis. All right. The fovea centralis contains only cones. So you ask yourself, well, what kind of light or what kind of colors? Well, all kinds of colors and not black and white, because the cone cells are the cells, let's say, that operate in bright light and help us see color. So the fovea centralis contains only cone cells. Therefore, we can see with great visual acuity, meaning we can see in great detail. I'm just going to write that instead of type it. Detail. Oops. Man, oh man, I can't write today. Sorry, guys. Fovea centralis. And actually, it's called the centralis because it's in the center of our retina. The macula lutea, in contrast, contains mostly cones, but there are rod cells as well. So as a result, that allows us to see in black and white. However, we should have already completed an activity where it allows for this is where our peripheral vision comes in. And what our activities that we demonstrated showed us that we don't have great visual acuity or color detective skills in our peripheral vision. It's because it doesn't contain only cones. It only contains some cones. And then finally, the optic disc is the disc that's actually, or it's the space that our optic nerve attaches to our retina. And because the optic nerve attaches to the retina here, there are no rods and cones. And because there are no rods and cones, we can't detect light there. So in the other activity with the note card that we completed on Thursday, where we were looking at the square with our left eye closed on the note card, and all of a sudden the circle to the right disappeared. It's because the circle to the right was being focused right on our blind spot. It was